This video is made in collaboration with the Canadian Ornamental Horticultural Alliance. With growing environmental challenges and water supply uncertainties, reusing irrigation water is becoming increasingly more important. But is that recycled water clean and safe for reuse? What are the major issues surrounding the reuse of irrigation water? A survey asking U.S. growers what contaminants were of the most concern indicated that they are plant growth regulators and pesticides, plant pathogens, and nitrates, phosphates, and salts. Hi, I'm Dr. Ann Huber of the Soil Resource Group. Our team has been doing research on treating greenhouse and nursery water to ensure that it is safe for reuse on crops. In this video presentation, we will briefly explain the design and benefits of customized hybrid treatment systems for greenhouse and nursery growers. Hybrid treatment systems could be thought of as enhanced constructed wetlands. They are not the vegetated wetlands you may be familiar with, but rather employ a combination of media with various physical, chemical, and biological characteristics designed to clean return irrigation water before it is reused on crops, and they can be designed to treat water to meet individual site-specific requirements. To test the design parameters for hybrid treatment systems, 2 portable pilot systems were built specifically for research purposes. Each tank or cell contains a different media, including wood chips, pea gravel mixed with slag, wollastonite, filter sand, or granular activated carbon. Contaminated water is run through a series of these cells, and the treated water is returned to the irrigation system. Our early research on hybrid treatment systems showed that they can successfully remove undesired components such as nitrogen, phosphorus, and fungal pathogens. For example, water coming into the system may be high in nitrate nitrogen. We call this water the influent. After going through the media sequence, you can see that the post-treatment water, or effluent water, has very little nitrate left. This graph illustrates that the amount of nitrate in the influent, the left bar, is decreased to below our target level after treatment. In this next image, you can see that the average fungal population is reduced to below detection limit after going through the wood chip cell of the system. This image of typical 3M testing results shows that reduction visually. We are now looking at these systems for their ability to remove plant growth regulators, also known as PGRs, and pesticides. Plant growth regulators are the primary focus of the study because, while they are powerful tools for crop management, it is important that recycled water does not carry residual PGRs to the wrong crop or to the target crop at the wrong time. So, can these systems remove PGRs and pesticides? We used both chemical analysis and plant bioassays to answer this question. And while the project is not quite completed, for most of the PGRs and pesticides tested, the answer appears to be yes. In our pilot studies, influent water from a greenhouse recirculation system was spiked with 4 PGRs to ensure we could properly test removal efficiencies. Water samples were collected periodically over the trial runs and sent off to the laboratory to test for PGR and pesticide residues. These samples were also tested in bioassays, assessing the germination and growth response of broccoli seedlings to any residual PGRs or treatment side effects that might not be captured in the laboratory tests. As an example, in our August series run, we tested the water after each media cell, and laboratory analysis indicated that the first cell in the treatment series, wood chips, removed over 99% of the PGRs, and 4 of the 5 other pesticides detected in the influent greenhouse water. The bioassays showed that plant growth was restored in the treated water, indicating that there were no unintended side effects of the treatment process. In this table, you can see that over 90% of detected PGRs and pesticides were removed by the various media we used in our batch studies. To remove all PGRs and pesticides present, we need to combine several media types in sequence. Of course, the portable cells used by the research team are too small to meet the needs of most grower operations. On a commercial scale, once an adequate sequence has been determined, large-scale in-ground systems are constructed. In these pictures, you can see the various stages of construction of permanent hybrid treatment systems. Following construction, the systems can even be used as a production surface, provided there is no machine traffic, of course, so no loss of production space. There are now 3 full-size permanent hybrid treatment systems installed in Ontario. So, in summary, if they are designed and managed properly, they can 1. Remove excess nutrients, 2. Remove fungal pathogens, and 3. Remove common PGRs and pesticides. However, their performance is impacted by factors such as temperature and flow rate, so proper design and management are very important. You can find more about these results at the COHA Connections research website or Flowers Canada Ontario resource page at the links you see here.